Hey guys, last couple of days in class we have gone over cellular transport. So I wanted to go over this with you today. Tomorrow I'll be posting a little worksheet that you can work on that and this video will hopefully help you complete that worksheet. And also, if you weren't in class on Monday, which several of you weren't, I would encourage you watching this video to help you understand, because this can be somewhat of a complicated subject. So first of all, we're going to talk about this concept of diffusion. Diffusion is just the movement of particles from a high concentration to a low concentration. We say that these particles move down their concentration gradient or with their concentration gradient. No energy is going to require it. So it's a slow process. It relies on the random motion of atoms and molecules. And again, particles in diffusion always go from high to low in their concentration. There's three key factors that affect the rate of it. The first one is the concentration. So the concentration of the substance, the higher the concentration, the faster the diffusion is going to take place. And as things start to spread out, it becomes slower and slower. Temperature will increase the rate of diffusion. If you have a higher temperature, those particles move around more, they bounce off each other more, and they move around and spread out more. And the last one is pressure. When you increase the pressure inside of an object or in the volume of a container, then the diffusion is going to occur more rapidly. There's a concept called osmosis that you may have heard of before. Osmosis is the diffusion of water across a selectively permeable membrane. So water always is going to move from high to low. So a high concentration of water to a low concentration of water. And so that's what osmosis is. It's moving across this membrane from high to low. And there's three key factors that are going to affect, I'll say three key factors. There's three things that we're going to talk about osmosis from 3 points of view, okay? So before we do that, there are three prefixes that you really need to understand and have a good grasp of. Okay, the first one is iso, and it means equal. And I think of an isosceles triangle with two equal sides. That helps you remember what iso means. Iso means equal. It's really the easiest one of the three that we're going to learn about. Hypo means under. You might think of some other words that have hypo in it. A hypodermic needle goes under your skin. Hypothermia, it means you have a below or low temperature. Hypoallergenic means that you might have a hypoallergenic laundry detergent or something. It means that there's low allergens in that. The word hyper, think about hyperactive, meaning you're overactive, above active. Or if you hyperextend your elbow, you've gone over or beyond where it normally extends to, right? So these are, or if you have hypertension, that's high blood pressure. And so these words will help you remember what the words hypo, hyper, and iso means. So the first one we're going to talk about is this concept called an isotonic solution. So we're talking about the solution that would surround a cell, okay, in an isotonic solution. In an isotonic solution, the concentration of dissolved substances, another word for dissolved substances that we talked about in class is solute. So if you have a solution, the solute is the dissolved substances that dissolve in the solvent, which is the liquid, okay, which might be water. So the solute or the dissolved substances in the solution is the same as the solute or dissolved substances inside the cell. So when they're both equal, that means the water is going to be equal, both inside and out of the cell. And so there's going to be an equal movement of water into and out of the cell. And so your cells in your body are in this isotonic solution. Now, some practical examples of that too, if you have to get a vaccine, whether it's the COVID vaccine coming up or maybe a flu vaccine, or you had a chickenpox vaccine when you were little, that is always going to be administered in an isotonic solution to your blood, because when they inject that in your arm, we don't want that to damage any of your cells. So we want to make sure that's isotonic to your blood. When you get an IV, it's the same way. That IV, whether it's saline solution or some type of sugar water, is going to be isotonic to your blood so that there is not a damage to any of your red blood cells. So that was the pretty easy one to understand. The other two are a little more complicated. So the first one is called a hypotonic solution. So if you remember, the prefix hypo means low. So this says the concentration of dissolved substances is lower in the solution outside the cell than the concentration of the substances inside the cell. Okay, so that hypotonic solution, that means that there's a low solute concentration outside the cell. Anytime there's a low solute concentration, there has to be a high concentration of water, okay, in that solution. And so remember, water is always going to go from high to low. So in this scenario, where's the water going to go? It's going to go into the cell. Now, what I've done here, you notice on the hypo I've made this big O here, and that will help you remember that in a hypotonic solution, water goes into the cell and it can swell up, and it can actually sometimes burst. So there's this net movement of water into the cell in a hypotonic solution. Hypertonic is hyper meaning high or above, so the concentration of dissolved substances or solute outside the cell is higher than inside the cell. So if you have a high amount of solute concentration, you means you have a low amount of water. So which way is the water going to go? There's a higher amount of water inside the cell than outside, so the water will leave the cell and it will exit the cell. So you can kind of remember that hyper with an E, water will exit the cell if it's in a hypertonic solution. And if you look at this picture here, in a hypertonic, these red blood cells will lose water, so there's a net movement of water outside the cell, and they will shrivel up and shrink and lose mass and all that. So that is a hypertonic. A couple of real-life examples that I gave in class. If you take freshwater fish live in a hypotonic solution, low solute, high amounts of water. So they have a tendency to take water in through their cells. They will just kind of diffuse into their cells. So their kidneys, their excretory system has to adjust for that. So when they make their waste, they have a very diluted waste. They have a lot of water that they have to flush out with their waste. So they have a very watery or diluted waste. Saltwater fish are just the opposite. They have a tendency to lose water to their environment because they're in a hypertonic solution environment. And so their kidneys have to work the opposite. They have to conserve water. So they have a very concentrated waste and a very dry waste. So if you were to take a freshwater fish and put it in salt water, and remember, they have a tendency to try to get rid of water in their kidneys, and then they're in that hypertonic environment, they're going to lose more water, and they will die from dehydration. You take a saltwater fish and put it into fresh water, it will, remember, they have a tendency to conserve water and hold water in, and then they come into an environment where they're going to be taking in a lot of water. They will have a tendency to overhydrate, and they would die from that. Another good example is if you have a sore throat, your mom might tell you to gargle salt water. Well, what's causing the sore throat is the swelling in your throat from the fluid buildup that's fighting that infection. So when you have this buildup of fluid or water in your throat, gargling with that salt water will help draw that water out and reduce some of the swelling. It also can help you by killing the bacteria. The bacteria would be exposed to that saltwater environment. They would lose water and they could die from it. So it's beneficial in that way. So a little bit about hypertonic, hypotonic, and isotonic solution. We're doing a quick experiment in class, a lab that you can do at home if you wanted to try. It's real simple. You take 2 eggs and you soak them overnight in vinegar over in 1 day. And that vinegar will dissolve the shell. And then you take one of the eggs that has just that membrane around it, and you can put it in just regular old water, submerge it in water, and leave it for a day. And you take the other and put it in syrup, in pancake syrup, and leave it for a day. So the water solution that you put it in is a hypotonic solution, and the syrup is a hypertonic solution. And then you'll notice a dramatic change in those 2 eggs the next day. So we're going to be doing that in here, and I would encourage you to do that at home if you have some eggs laying around, and mom won't care. And it's a good way of visualizing these hypertonic and hypotonic solutions. Today in class we talked about passive and active transport. So passive transport is the movement of particles with or down their concentration gradient. So I like to think about floating down a river. If you float down a river in a canoe, it doesn't require any energy. So you just go with the current. So passive transport does not use any energy. Some examples of that are diffusion. Diffusion is whenever particles will just diffuse across that membrane from a high concentration to a low. It's important to realize because of this nonpolar region of the cell membrane that only small nonpolar particles can pass through that membrane. So I'll include that in your notes. Small and nonpolar. Example is oxygen. Oxygen is small and nonpolar because it's O2, two water oxygen molecules bonded together. It can diffuse into the membrane. Anything that is polar or larger have to go through these proteins. So this is a channel protein that water, for example, will kind of just go through a channel protein and go from a high concentration to a low concentration. Carrier proteins can also carry things into the cell from a high to low. This is what we call facilitated diffusion, or diffusion that is helped by these proteins. So the key to passive transport, again, that there is no energy required. Active transport is the movement of particles from low to high against their gradient, or up their gradient. So it requires energy for this to take place. And it occurs through these carrier proteins. And if you look at this, the example I like to give is of a vacuum cleaner. Vacuum cleaners take dirt from a carpet, which is in a low concentration, and put them into the vacuum bag, which is in a high concentration. So it requires energy. You have to plug that vacuum in to get it to do that. This is an example of the active transport. This is what we call the sodium-potassium pump. It's a classic example of active transport. We were actively transporting potassium into the cell. So potassium would be in a low concentration, these little triangles. Out here we actively pump it into the cell. And the sodium is in a low concentration in the cell. We're going to actively pump it out to a higher concentration. So ATP is going to be required, which is cellular energy. And the last thing that we didn't get to go over today, but we're going to cover it tomorrow real quick, is these two terms, endocytosis and exocytosis. Endocytosis is the process by which a cell surrounds a substance, encloses it with its plasma membrane, and brings it into the cell. So the cell's kind of like this. White blood cells are the classic example that do this endocytosis. They will surround a food particle or maybe a bacteria or a virus and then just bring it into the cell. They engulf it and bring it into its body to break it down, digest it, or whatever they want to do with it or consume it. That's endo, so you're going into the cell. And exocytosis is the opposite. That's whenever waste or proteins or things that need to be secreted from the cell, large particles, the cell membrane's here like this, and the particle will come up here and it will release it outside the cell. So it doesn't actually cross the membrane. It actually gets engulfed and brought in and kind of pinched off and let out of the cell. So that's a little bit about active transport. I know I kind of went through this pretty quickly. Hopefully you understood it a little bit. Give me an email if there's something you don't understand. The worksheet tomorrow, feel free to ask me any questions you have on that if there's something you don't understand if you're not in person. So I am here. Now we'll be doing a test on Friday over cell transport and the cell cycle, so be looking for that as well. And then next week we'll have our semester test. And more information will come down about that later. Okay, thank you. Let me know if you have any questions.